Good afternoon, everyone. Welcome to our flight to challenging a dogma. After you have all put your mobile phones on flight safe mode, we can. I hope that the minority of you will feel those side effects described here, but that the majority of you will help me to challenge a dogma. And the dogma here is: don't believe that something is safe if someone tells you that. And this is especially true if we are talking about biosafety. Today I will only be able to give you a short introduction, because I could talk weeks and months about biosafety. I can tell you a little bit about my personal background. I'm an educated virologist from the University of Copenhagen, and I'm working as a researcher here at the Faculty of Science 2008 now. And I have made some experiences with biosafety in Norway. And the point with biosafety is that you can do things in the right way, in the right way, or in the wrong way. And if you do it in the wrong way, it's sometimes really shit. So what I try to teach you today is to do things in the right way, or to learn how things can be done in a correct way. And usually I should have this. Doesn't work. No, it's working. Okay. The first time I've been, as I have moved to Norway and looked on the biosafety system here, I was a little bit astonished. The European standard is that biosafety is handled by a microbiologist, at least at MSc level, for the whole faculty. Here, biosafety is either belongs to the responsibility of a technician, a senior technician, to the human resources administration department, or to the project leader. The senior technician or technician can have huge experience, and I have a lot of respect for those people working with biosafety there and having biosafety as their responsibility. The problem is, you don't know if the person who is responsible has a clear idea what biosafety is, what their tasks are, and you very often don't know if they have actually the skills to doing their job. The project leader, you could think about that the project leader, okay, is a senior scientist, he or she should really know what they are doing. I had a case where a bioinformatician was the project leader and a student working with a cancer-causing virus. A bioinformatician has not the skills to handle such a project with regard to biosafety. He or she has the skills to be a good scientist, but with regard to biosafety, a bioinformatician has not the skills. So this is a problem here. And I hope during the years this will change here. Also, molecular biologists have generally no training to biosafety. You can have an excellent researcher, can have excellent molecular biologists, but if they don't have the training to understand the risk which is associated with a biological agent, you have a problem. You have it. Because it's your health, it's your safety. And I have had a discussion with the person responsible at the largest institute here for biosafety, and she was telling me, yeah, we are working only with viral vectors. Of course, they are biosafety class 2, but we are only working with vectors, not with the virus themselves. Okay, but adenoviral vectors, which are very often used, can make your eye looking like this. Epstein-Barr virus. Many of you have already worked with cell culture, I expect. Cell lines transformed to propagate in a never-ending timeline. They are very often transformed by Epstein-Barr virus. If you have a cell line which has gone into 70s, it's okay. The virus is integrated, and there's no harm from this particular virus. If you have ordered just recently a new cell line, which has gone only 1 or 2 passages, so which has only propagated 1 or 2 times, then you still have active virus. And this particular virus can, in rare cases, cause cancer. And so you have to be aware of the potential risks. And it's a little bit to be or not to be. Yesterday some of you have heard that we have biosafety cabinets. If you work wrong with those cabinets, they will not protect you. If you're working with blood or with any other tissue samples, if you're working with hepatitis C, look on this picture. If hepatitis C would attacking your face instead of your liver, you would do something about it. So it's Hantan virus. We have several agents. I'm personally working with this particular virus, which is within a BSL-3 facility. I will later on explain what BSL-2, BSL-3 is. You have all the time be aware of the risks. And one important point is there's no cabinet, no procedure that alone guarantees your safety. You have to think. You have all the time to think, what the hell am I doing? And you have actually to understand what you're doing. I guess you have been around for the lab center. Some of you are already working at St. Olaf, at some places I know. But you will see nearly on each floor this sign. How many of you know what it means? 1, 2, 3, 4. So this sign means danger. Danger. There's something in this room which makes you which has the potential to make you sick. Be aware of that. You will sometimes see that people are bringing their children within those labs. On the doors is standing this sign. And the majority of the people don't understand what this sign means, this sign. So, biohazard is something what has the potential to make you sick. And if you're looking on a laboratory, what are you doing in a lab? You're not doing work like an industry, things which people have done thousands times. No, in a laboratory, you're doing unusual things with potentially hazardous material. So sometimes you're doing procedures which have been performed 1,000 times. I have more than 10 years experience in a laboratory, in a high security lab, and I was all the time used to take samples out of nitrogen tank, which means that it has been cooled down to 192 degrees, where viable virus is inside. In that case, it was HIV. Then I have done that 1,000 times. I've taken it out of the nitrogen and was thinking about something else, and then the tube exploded and I was over and over spilled with HIV. Luckily, I was not become infected because this virus is not really good transmitted by air. But Either you're doing unusual things, or you're doing things 1,000 times. Never ever stop thinking. And even experienced people make this error, that they are not thinking about the procedure they are doing. If we are looking on the literature, and those are only the cases which are reported by the literature. You have nearly 4,000 cases within the last year in 5,000 labs. The last one, you may have heard about the hemorrhagic E. coli infection last year in Germany. There was a lab technician which has become infected in the lab with a bacterium which potentially can lead to hemorrhagic disease, so you're bleeding in hell. Be aware all the time of the risks. And why? People have studied on this long time, and they have found some principles, some red line which has gone through all those 5,000 lab accidents. The first one, the lack of instruction for handling infected material. Then, the removal or not existence of trained workers, trained technicians, trained scientists on biosafety. New diseases have come up. We are working with gene technology. We are changing the genes of pathogens. We are changing the genes of potentially harmless bacteria, viruses. We don't know what actually is going to happen with them. Lack of protection of workers. Sometimes people think the white lab flag is a status symbol and not a protection, a thing for protection. You will sometimes see people running around, not sometimes, very often see people running around in the canteen and afterwards going to the patients. And what we don't have, and that's a problem, we have an inadequate system of inspection of the premises. And that's clear, of course, if we have our system based on human resources, they have not the skills to make inspection. They're not even thinking about it. Laboratory safety is not only for you. It's also for the people around you, for your husband, for your wife, for your friend, for your children. This was a case where a person has been vaccinated in a research lab from the US military with a vaccinia virus. His family members, this is the soldier, and his wife have also become disinfected. It's not very nice to have all those variola-like, pox-like clumps in your face and your inner organs. The same again has happened also with this hemorrhagic bacterium in Germany last year, that the family member has become infected. Some of you are going to work with neuroscience. It's very common that you will use rabies, viral vectors based on rabies virus, because they are going in the brain and they are the perfect proper tool to do it. And most of the cases it's also safe. But think about it, if you would have in your viral vectors, rabies viral vectors, one viable virus which can infect not only you but also your children. Be aware of the risks, understand the procedures. What we can do, or what biosafety is about, is for protection and not the product, not your experiment. It's for protection of you, your co-workers, the people cleaning the lab, the environment, so all the people around you, and In the very last thinking, it's about protecting your product. But don't think about it. The problem is that the majority of people are thinking about protecting their product. They have worked weeks and weeks on their molecular biological experiment. They have worked weeks and weeks on the animal experiment. And then it has to work. Otherwise, I will not earn my degree. So it has to work. And then they are stopping thinking what they are doing. So think about biosafety. Think that you have only one life, one health. So the principle within biosafety is, the goal is to reduce the risk. And please remember, it's not only infectious disease people like me who have to think about biosafety. It's everyone working with living organisms or with recombinant DNA research in a lab. And the majority of you will do that. So there is a risk. We have to think about the risk. And if we are calling about the agents that would create the risk, and there's a risk to the workers, to you, to your co-students, to all the other people in the environment, but you are very often not sure what the risk is. What will this inclusion of this certain piece of DNA to your bacteria or to your virus, what will result out of it? So you have to think about the risk and you have to determine the risk. No risk can be achieved, of course, if you're building your lab on a Mars. And but that's not what we are doing. All of you are dreaming to become scientists. Why? Not the salary, not the career path, but because the majority of you will change something. Will contribute to the development of the human race, call it that way. But the majority of you will do something. So we have to live with our risks, and we have to understand them, and we have to minimize them. But remember that there is always a risk. If someone told you, doesn't matter since when he or she has been professor, how many publications he or she has, if someone tells you that there is no risk, it's a lie. There is always a risk. How small it may be, there is a risk. But we have to accept, in our job we have to accept this risk and we have to try to control it. And with regard to biological hazards, the most important thing is that you know your agent of risk. You have to understand the biology behind it. You have to understand how this virus, this bacteria can, or cell line, can make you sick and in which way it can enter your body. You have to understand which risks are you accepting when you are pipetting. Even there's a risk. So what you should do before you're starting, think about a worst case scenario. After you have understood what you are doing, after you have read about your biological agent, think about the worst possible thing that can happen. Think then about the likelihood, the seriousness. And then what can you do to reduce the seriousness? What can you do to reduce the likelihood? And then do it, of course. So, but all the time think about there is always a risk. You have to judge what is acceptable, what you can accept and what not. But who determines the risk? There's all the time also an interest in crazy people working with crazy diseases. Therefore, each of you has to think. Do not stop thinking if anyone tells you this is safe. You have to think it. Because we all have our interests. We want to have the new research money, we want to have our degree, we want to have this paper published. Okay. But remember the risk. One thing to reduce the risk, what we scientists like to do is to classify things. One way of classification is a biosafety level. The European Union has its own definition. It's very similar to the definition which the US has. But a biosafety level is the level of biocontainment precautions, so the things you can do to reduce the risk required to isolate dangerous biological agents. There's written dangerous. I don't like this dangerous because it would be much better to have biological agents. Because this implies that you have also biological agents where there is no risk. But because we are all doing unusual things, there is always a risk of a potential dangerous agent. So what we have, or what the European Union, even if Norway is not a member of the European Union, they follow their guidelines, mostly. So we have 4 levels. Level 1. If you ever hold a presentation later on, don't make a slide with so much text, it's not good. Biosafety level 1. Where you are going to work with well-characterized agents not known to consistently cause disease. So they can cause disease, but not to everyone, not everywhere. Level 2. The most of the labs, the majority of the labs here are biosafety level 2 labs. And this means that there's a moderate potential hazard to the personnel and the environment. Environment, remember your family? So, and there's, for example, the different influenza strains. 2 years ago, we have had the so-called swine influenza. It's actually a bird virus, but even you are allowed, those of you working at St. Olaf have all the time been in contact with patients having this virus. Even if it's classified as biosafety level 2, this particular type could have been biosafety level 3 or even worse 4. So understand the biology of the agent you're working with. Biosafety level 3. We're having 1 currently in working at the NTNU, at the medical faculty, and 2 are going to be built now. So in biosafety level 3, you're working with agents that can lead to serious disease or that can kill you. And this may be the hantavirus I'm working with, the one with a lot of blood in the beginning of the slide, dengue virus, yellow fever, West Nile, Rift Valley. So, if you're working in such a lab, remember this can kill you. You're making the error only one time. Biosafety level 4, Norway doesn't have one. So we will not start thinking about it. But those of you who have, I think Contagious, it was called, and Outbreak with Dustin Hoffman some years ago. There have been a lot of movies about biosafety level 4 labs. There is 1 in Sweden, 1 in Denmark, and 4 in Germany, closest to us. But there the people are working with Ebola, Marburg, pox virus, the really bad ones. We are not working about it, therefore I will not talk much about it. Okay. Biosafety level 1 is suitable for well-characterized agents not known to cause disease in healthy adult humans. There's nothing standing about pregnant women, there's nothing standing about children, there's nothing standing about people with HIV. So it still is a risk, even not for the majority of us. What do you need for running such a lab? Biosafety level 1. You need a knowledgeable supervisor. This is not defined what a knowledgeable supervisor is. You need people who know something. You need to be aware of the potential hazards, and you need to know how to do things. And there should be a biosafety manual. I can tell you in most of the labs here, designated as biosafety level 2, you will not find a biosafety manual. The only thing you will find is this blue book, which the majority of you have not read yet. Okay. The building, the lab, has to have walls and things like that. I think that was what I was saying about this slide, I don't remember. What is important is that you have the tools to work safe. You need to have biosafety cabinets. You need to have protective clothing. And the protective clothing, again, requires that you understand what the risk of transmissions are. Pipetting device. Never ever do it with the mouth. Bacillus subtilis, E. coli. E. coli is usually biosafety level 1 bacterium, but if it's infected with certain bacterial viruses, it can make you bleeding to hell. Think about that. Even this is not physics, it's not exact science. You're working with biological entities. Oh. Yeah, exactly, yeah. If you believe that the law all the time will protect you, believe it further, but it's not the case. If you look on the minimal requirements regarding to the law, is a biosafety level 1 lab needs to have doors. That's regulated in the lab and in the law. You need to have the possibility to wash your hands. The surface should be easily cleaned, and the bench tops can be easily decontaminated. This is a biosafety level 1 lab. Okay, the location doesn't need to be separated, the structure needs to be a normal structure, and there's no special requirements for the ventilation. Standard microbiological practices. Restrict or limit access when working. You will not see this. You will sometimes see people bringing their children in BS2 labs. Prohibit eating, drinking, and smoking. Each of you will think, of course, no one of us will do that. How many of you have a chewing gum now in their mouth? Say 1, 2, 3. How many of you will remove it when you're entering the lab? Not many. This is also eating. Because there is a likelihood that you bite yourself with having a chewing gum. And then you're working with something strange. And then you have an open wound in your mouth. And then you have done it in the wrong way and your hand's full of shit. Don't do that. Even chewing gum is eating in the lab. Okay. How many of you have ever seen someone mouth pipetting? Wow. Okay. Don't do that. Wash your hands. Even if you're leaving a toilet, you should wash your hands. Yes, please?
Sir, like I want to ask a question about, you say that whenever you have a chewing gum in your mouth and you're entering the lab, that's also a contamination, kind of a possibility of contamination, right?
Yes.
But I've seen some people like who are very, very higher scientists and they're working in biosafety level, they don't give a damn about that. And they used to eat also, they used to drink. So it shouldn't be like that. There should be a kind of a proper investigation about that, that the people who are like junior and working as a co-workers and something, they should not be, like with them, also the scientists and the.
I absolutely agree. And you will very often see high-ranked scientists doing stupid things.
Okay.
And one in the US, I think there's a proper system. Many European, there's a biosafety standard which is applied to my knowledge, to Germany, to Sweden, to Denmark, to Belgium and the Netherlands currently. It's not applied in Norway. And with regard to your question, have a look on Janteloven, the law of Jante, which is a special problem in Norway, which essentially means you don't have to tell me something because you're too small to tell me something.
Yeah.
That's a problem. I absolutely agree. That are stupid things, and you will see high-ranked scientists doing stupid things. To have an academic degree doesn't mean that you're all the time clever.
And I have a kind of an experience where I see that before the evaluation, people are coming. They are informed that evaluation people is coming, so they get everything right, and everything seems normal. And so, like the evaluation procedure should be more stronger.
It should be more stronger. It should be happen at all,
Yes.
and it should not be expected by other people. But the point is, it's about yourself. Biosafety is about yourself and your family. It's not for satisfying the director or something else. It's for you and your family. And washing your hands. A lot of people are leaving the toilet without washing their hands and contaminating a lot of things. Protective clothing, lab coat, clothes. You have yesterday heard that there are many, many, many different types of clothes. And the blue bag, the blue book, which you haven't received yet, that's also a shame, actually. There is a list about potential clothes and use of clothes, but you can Google it and find it. Okay, yes. Sometimes you need your eye protection, sometimes you need a face shield, but it depends all the time on the procedure. And think about if you're working, for example, with a biological agent, what will this face shield protect you against? What's the most dangerous thing you can do when you have your hands on a potential lethal virus bacteria? What's the most dangerous thing you do?
Like you want to know the name of that?
No, I want to know the proceeding, the movement you can do.
You can wipe your face.
Exactly. And that's what the face shield is protecting you against. Because you very often, think about, observe yourself now, how often you touch your face, your nose, your eyes, very often. So therefore a face shield is to protect you against yourself actually. Special practices in the BSO1 lab now. According to the law. The training requirements, the supervisor should be a scientist with general training. It's not stated in the law what general training means, so you can end up with a bioinformatician as a project leader telling you what you should do in a microbiology lab. The lab personnel should receive specific training in the lab procedures. But who is training the trainers? Also think about it. Biosafety level 2. The majority of the labs we're having here at the lab center or on the campus. It's suitable for work involving agents of moderate potential hazards to yourself, to your colleagues, to your family. Biosafety level 2, moderate. If anyone tells you it's safe to work, we don't have to look at this and this procedure because we are only working with biosafety level 2, forget it. You have to remember that biosafety level 2 means moderate potential hazard. You will not die of it, but you will not like it. The requirements by law are that the laboratories have to have lockable doors. Of course, that there is a sink for hand washing, surfaces easy cleaned, study furniture, not like this will not work. Biological safety cabinets, light, airflow into lab without recirculation in non-lab areas. Go through all the labs, have a look how many are directly connected to office floors. And regarding to the law, windows fitted with fly screens also. Restricted access when work in progress, of course. You should have the possibility to decontaminate your tools. The location should be separated from public areas. The structure can be normal structure and the ventilation should be directional. Should use standard microbiological practice. You should of course know what standard microbiological practices are. Majority, I will later on tell you a little bit more about biosafety cabinets. So, but the main rule, use biosafety cabinets as much as possible. But especially if you're doing handlings, if you're doing work procedures where you can produce a lot of aerosols. What is the most aerosol producing procedure in the laboratory? What do you think? Pipetting. Whenever you're working with large volumes and high concentrations of an agent, work in a biosafety cabinet. Don't have it look like my desktop in my office. You need to have order on your biosafety cabinet benchmark. This picture is actually much too much. It's in a textbook about biosafety and should show how a good organized cabinet should look like. This is far too much in the cabinet. And sometimes hard to work in a biosafety cabinet. Okay, still biosafety level 2, but this is valid for all labs, all lab work. Needles and sharp things, scalpels, whatever, should come in extra sharp container. Don't break, don't resheet needles, put them there for one time use only, put them in sharp containers, otherwise people working with the trash you're producing will come in serious problems. Don't touch broken glass with your hands. Something you should not do at home, but for sure not do at a lab. Whatever lab you're working in, use plastic as much as possible. carcasses and body parts. Know what you are working with. Animal parts, tissue parts, label them correctly, and treat them. When you're thinking they are infectious, treat them as infectious. And then they have to be handled accordingly. Special practices in a biosafety level 2 lab. Policies and procedures for entry. The policies and procedures for entry here for biosafety level 2, you will come with this card you will receive, or you have in most of the labs here. You need to have biohazard warning signs on the lab doors. You will see they are very often there, but again, how many know what it means? You need a biosafety manual specific to the lab, and you need training with annual updates. This is the first biosafety course given here for master students, to my knowledge. Depending on what you are working with, you should have baseline serum samples, so once a year or once every 6 months. You should have a look if you have become infected, and if possible, you should be immunized. After work, decontaminate the surfaces. Report immediately spills and accidents. No animal, and please no, never ever children in the lab. The supervisor is a competent according to the law, a competent scientist with increased responsibilities. Means also everything and nothing. And in biosafety level 2, there should be limit access if people are immune compromised. Are you immune compromised if you're pregnant? Yes, you are. The people working there should be aware, really aware. They should know what they are doing. Biosafety level 3. Suitable to work with agents causing seriously or potentially lethal disease. You will die. Tuberculosis, encephalitis, hand-touch. There are many viruses and bacteria which could have it. If you're looking on the law, it should be in a separate building, one isolated zone, should have been a double door entry, and there should be an air change, so that only air comes in and no air comes out without going through a filter. The human penetration should be sealed. The walls, floors and ceilings are water resistant, and it's easy to clean them. And the lab workers should clean them by themselves and not letting cleaning personnel in which don't have received the proper education. Whenever you are working with cell culture, which could have been, and this is true also in my opinion for biosafety level 2, what I'm telling you are the minimum standards according to the law. You should every time use also HEPA filters. HEPA filters as particle filter, which exactly, and it should remove most of the potentially dangerous agents. All the time work in a biosafety cabinet. The right one. Use respiratory protection. Be aware that you use proper standards. Plus that your tools, your biosafety cabinet, is controlled, that it's working. It doesn't help if you have one standing there and it's not working. You have to wear double gloves when you go in. One short point with regard to the gloves. Latex gloves are protecting against microorganisms. Nitrile, the standard nitrile, are protecting against chemicals. Take this as roll over the thumb. And wear triple gloves whenever you're really handling an infectious agent. Supervision here, according to the law, supervisor should be a competent scientist, experienced to work with the scientist. This is the only time in the law where it's stated that the supervisor should know what he or she is doing. You should have criteria established for the entry, and you should train the lab personnel. That's the most important thing. And the lab personnel should follow the guidelines. And of course, be honestly and report each incident which may happen. Biosafety level 4, this is the one I have worked in Denmark in. We don't have it here, so we will not talk about it. So again, this is a short overview. You have biosafety level 1, where you have good microbiological techniques. This is also true for molecular biologists, not only for microbiologists. Neuroscience, whatever. Ever you are working with cell culture, it's also microbiology. You can work according to the law on an open bench. It doesn't need any safety equipment. Biosafety level 2, you need to have protective clothing and a button on your door. You can work open on your bench, but you should have the possibility to work in a biosafety cabinet as well. Biosafety level 3, control access and directional airflow, very important. And nearly everything should be done on a biosafety cabinet. Biosafety level 4 we don't have here in Norway, and that would be a history for itself. Okay. Biosafety cabinets. As I told you, it's actually on this slide it's written in the wrong order. Because the purpose of the biosafety project is the most important purpose is to protect you against the agent of risk, to protect the environment, and last priority, to protect your product, to protect your experiment. You will see that there are existing 3 classes of biosafety cabinets. Class 1, forget it, it's not a biosafety cabinet in my opinion. Class 2, most of the cabinets you will see here at the university are class 2 biosafety cabinets. They are sufficient for the majority of the experiments. Class 3, they are very secure, but it's very difficult to work with those. And we have only one. Okay, class 1. They can be with or without a particle filter, a HEPA filter. The majority of the air, if there is a HEPA filter, it's okay. Then it's at least filtered, the air which comes out. If there is no one, but it can still be called biosafety cabinet. Without HEPA filter, everything goes out, and it's mainly only for product protection. Biosafety cabinets class 2A and B. Majority we have is class 2A. And you see here in red it's written 30% is exhausted, is released to the room where you're sitting. However, nearly 99.9% you will have a construction above the cabinet, which removes the air and which goes directly into another HEPA filter. So I would say that those cabinets are safe, plus that you have all the time, if you're looking back to the class 1, the red one is the contaminated air, potentially contaminated. So yeah, in the class 1 you have all the time contaminated air which is not removed. Each time you're putting your hands in, you're putting it in contaminated air. In class 2, both 2A and 2B, it has at least gone once through the HEPA filter, so it should theoretically be clean. The class 2B, I don't believe that they are existing here in the house, but they have a 100% exhaust to the room. But the majority, again, are 2A. Have a look on the signs and the labels on the biosafety cabinets you're working on, if they are suitable to work with. This is class 3. We're going to thick rubber, not boots, rubber clothes, and where you can handle infected animals and really hard stuff, and it's really, really difficult to work with those ones. Okay, HEPA filter. High efficiency particulate air filter, so it traps particles only. Each time you're working with chemicals which gas out, it will eventually destroy your HEPA filter. Be aware of it, and it will not protect you against potential dangerous gases. The HEPA filter are usually metal or wood framed, and if you open one, it looks like this. And usually you don't need to come in contact with the filter itself. But again, the largest institute here at the faculty, we don't need to sterilize the filter, the bench, before the technician is opening it. This is stupid, this is wrong. It's stated in several biosafety manuals, it's stated in several European and World Health Organization standards. Each time you're going to open the HEPA filter, removing it, remember all the dangerous agents are collected there, you have to decontaminate the filter. You can do this with formaldehyde gas, and it's safe. If you say, no, we don't do that, we don't need it, we're working only with biosafety class 2, get it, it's wrong. If you're going to work with, and those of you who will go to work with cell culture, think about what you're going to do. Put everything on place before you start working. Put your hands straight in, and don't in, out, in, out. And think about the procedures you're doing. And decontaminate as soon as something happens, and decontaminate as soon as you're removing your hands out of it. Yes, safe operation. No sweeping motions, and watch that the cabinet is working. If you're working on a biosafety cabinet, this is from this house, remember why you are working in a cabinet. Because to protect yourself. And it's not clever to put the waste outside and to believe that the dangerous agent is removed as soon as you put it in the waste. People making this, then they contaminate the whole room, and putting themselves and your colleagues in danger. Before you are taking something out of the cabinet, decontaminate it. And remember why you are working in the cabinet. Also from here. The way the cabinets are working is that they have an airflow in front of you, protecting you from that air is coming out. If you're putting your flask in, or if you, like here, using the grill as a stative, you're destroying the airflow and there's no protection for you or your environment. Work alone. There's people who have come long further than you in your education. Then do that. Remember that chemicals can destroy the HEPA filter and you will not recognize it. And I once experienced that one person has sprayed everything with 70% ethanol in the cabinet because he, as it's correct, tried to decontaminate it, but the ethanol was still recirculating, and then he started to started to work with a Bunsen burner. Everything exploded. Remember that. Centrifuges, also something what you for sure will be used. If a centrifuge looks like this, leave the room and don't work with it. But sometimes mechanical failure can happen, something operational failure can happen, and if that happens, there's a risk of contamination. Because whatever there was in these tubes before, it's out in the air. So check the tubes, the centrifuge tubes, balance it accordingly, seal the tubes if necessary, ensure that everything is fitted as it should be, and close the lid during operation. Most modern centrifuges will not allow you to start them before you have closed them, or will not allow you to open it before they really have stopped. But if there has been a crash, then the centrifuge will stop, and then the centrifuge will allow you to open it. But most centrifuges have a small window where you can look in, and you will for sure hear it if the centrifuge crashed. So I think also that there's a potential danger with that. If you have small spills after breakage, remove them immediately. Decontamination. We have now first to define some words. Sterilization is the use of a physical or chemical procedure to destroy all microbial life. And no, this screen is there. Disinfection, I have to read those definitions because I don't remember them correctly. The use of a physical or chemical procedure to virtually eliminate all recognized microorganisms, but not spores. Antisepsis. That's what you very often before an operation will get on your skin. Should destroy or at least reduce all microorganisms. So, if you wish to decontaminate something, you have to think what several factors. You have to think really how much do I wish to sterilize? Do I wish to make only the area antiseptic? What do I wish to do? What is the thing I would like to treat? Is my skin? Then I could not use harsh decontamination chemicals. How easy is it to use? What's the price? How safe it is? It doesn't help you if you destroy a potentially cancerous agent, cancer-causing agent with a cancer-causing chemical. Also, think about it when you select the agent. What is the type of agent you are working with? How much? And is it potentially protected within other material, within cells? And how much time? There are really many things to think about. In general, you have three methods to decontaminate something: with heat, with chemicals, or with radiation. Heat is the most effective method. That's what we're doing in an autoclave. And that's so-called steam sterilization. But you have to be sure that it's working. It doesn't help if you steam sterilize something and it's not working. And that means also don't, at the end of a working day, don't put kilos of lab material, lab waste in one autoclave. It will not work. It will not be sterile. It will not be decontaminated. Tri-heat sterilization. That's important if you have body parts or something like that. You can not use it on all materials if you want to reuse those materials. But as I told you, it's mostly used in connection with body parts and animal carcasses. Chemicals. That's what we are going to use in a lab day, usually, and gases. But there were 14,000, or there for sure more. As I looked up in the literature, it was about 14,000, but this is some years ago. So there are a lot of chemicals around. Read what is written in the instruction. And have also look on those three abbreviations: HLD, ILD, and LLD. In the common lab, we're using the intermediate level disinfection chemicals. The high level disinfection chemicals, they are used if you have to do with bacterial spores, with agents which are highly resistant. You don't need it all the time. What the majority of the people are using is Virkon S, and as far as I know, it's the most famous or most often used chemical intermediate level disinfection chemical in Europe. And then you have the housekeeping disinfection chemicals. But disinfection does not replace that you work in a proper way. Okay. We are all going to do fancy DNA gene technology, dreaming about it. Just to remember what gene technology, recombinant DNA technology is. You're putting something what is not belonging to an individuum in the genetic code of an individuum, or an animal, or a cell. So you clone something which you're doing something what the nature is usually not doing, or where the nature needs a long time for doing it. That's what we are doing in recombinant DNA technician. Please remember again that generally a molecular biologist is not trained in biosafety. But that was the story with the professor who's doing stupid things. Can be excellent molecular biologist. So if you are working with expression vectors later, think about if that the piece of DNA you're putting in your genetically modified organism may increase the virulence, the potential to make you sick of it, or it may produce it. You have sometimes genetic parts, the messenger RNA of some viruses make you sick without being in a viral capsid. So even if you're working with DNA pieces from pathogenic microorganism or organism, remember the way of transmission. Some gene products, the products of the gene, code for toxins or have pharmaceutical effects. Viral vectors, more and more people are going to work with them. You have vectors are produced, you can buy them from chemical companies. Usually they are safe. And what they have done is they have taken wild type virus, removed the majority of the gene responsible for propagating it, and generated some areas within the viral genome which allows you to put DNA in it. So, but sometimes people are producing it and people can make errors. And sometimes there may be contamination or not a completely destruction of the wild type virus. Sometimes there may be the potential that the replication incompetent vector gets a modification in its genome which allows him to propagate again, to be a wild type virus. The likelihood is low, I know, but it exists. If you're working with transgenic animals, Dolly is the most famous example. You're changing the gene. You're putting something foreign in the gene and you don't know actually what will come out of it. If you remove parts of the gene, that's normally considered as not hazardous. It's considered, but nobody has proven it. So you can have, you're changing the expression of proteins what the cells are producing, and that can end up that you're producing toxins without knowing it, allergens, hormones, whatever. So each time you're going to change some genetic material, think about those questions. Is there an increase of infectivity or pathogenicity when you, or the potential for an increase? Could any disabling mutation within the reset be overcome as a result of the insertion? Don't know it. Does the foreign gene code for a pathogenic determinant from another organism? If something happens what you do not wish that it should happen, but if it happens, can you treat it? Can you eradicate it? That's the big discussion with gene technology in plants. Okay. Emergency procedures. If you stick yourself with a needle, with a scalpel, or whatever, somehow come in direct contact with an infectious agent or potentially infectious agent, stay calm. Don't panic. Clean the wound, the eye, the mouth, as soon as possible. Report to the medical clinic. The infectious disease department here is very good, but you have to be fast. That's really important. If you have worked with blood sample, which could have been infected with HIV/AIDS, you have 2 hours to apply treatment that you don't get AIDS. Be fast, don't hesitate. And then inform your supervisor. If something happens not with yourself, but you splash something in the lab, tell it to your co-workers. Say, hey, there's something around that may be dangerous. And then leave the lab. And then make sure that nobody entered the labs in the time where you're not there, and then notify your supervisor. Again, think about the potential reasons for accidents. Working disorganized, working too fast, working the whole evening without sleeping and eating, thinking that what you are doing is not dangerous, thinking that the biological agent is not living anymore, and also not living doesn't imply that it's not dangerous, and using protective equipment which is not working. That's it. Thank you very much.